Hi, I'm Larry Johnson, a professor at Texas A&M University, and today we're going to be talking about the nucleus and mitosis. We want to characterize the interphase nucleus, including the cell cycle, the nucleolus, chromatin, nuclear envelope, and the nuclear cytoplasmic interactions. We also want to characterize mitosis, including the structures with definitions of mechanism of chromosome movement and cytokinesis. Thank you. Now, if we remember the proposed evolutionary involvement of the cell membrane in organelle formation, you remember the prokaryotic cell moved into the ancient eukaryotic cell, bringing in the cell membrane around that and making two membranes in the mitochondria. A similar type thing, that's in the eukaryotic cell. A similar type thing is in the proposed evolutionary pathway of nucleus and endoplasmic reticulum, and that is the plasma membrane or the cell membrane enfolded in around the DNA, which there is DNA and there are ribosomes in prokaryotic cells, surrounds this and brings in the double membrane, and is why you have the center part of the nuclear envelope is continuous with the cisternae of the rough endoplasmic reticulum. So if we look at the nucleus, this is a nucleolus, this is a nucleus, and we can see this centrosome here with radiating microtubules. Also, there's intermediate filaments that's associated with the nuclear envelope. We can see nuclear pores in through here, the inner nuclear membrane, the outer nuclear membrane, making the nuclear envelope as you see. Intermediate filaments on the inside as well as the outside. This is the nuclear lamina or the fibrous lamina in through there, and that has intermediate filaments associated with it as well. And so the intermediate filaments help stabilize the nuclear envelope as they're on both sides of the nuclear envelope. So the nucleus is the archive of the cell. Here we see the nucleus of a cell, the various organelles, the cell membrane there, nuclear pores in through there. You see ribosomes on the outer component of a nuclear envelope. You can see the coil DNA inside. It's the archive of the cell. It's the nucleus encodes for all proteins and secretory products of all the cells in the body. All somatic cells have the same codes regardless of whatever function, nerve, epithelium, connective tissue, muscle, they all have the same information inside there. What's different is the expression of them that makes a nerve cell different from epithelium. Now, if we look at the nucleus, we can see the nuclear envelope. We can see some lighter areas and darker areas. And the dispersed area, lighter areas, is known as euchromatin. Euchromatin, we see that here in this nucleus. And then a lighter area we see there in this nucleus of this cell. Now, the darker substance, usually especially associated with the outer component in through there, and through here is the heterochromatin. That's the condensed part that's shut down much of its activity associated with whatever type of cell that is. And and the heterochromatin, euchromatin, differs with the different types of cell, a smooth muscle cell, a nerve cell, or the different types of cell. And so cells of an individual have the same genome, no matter if it's a monocyte, no matter if it's neutrophil, red blood cells before they leave their nucleus, all of them have the same genome. What's different is the expression of the genes, and it expresses it by its dispersion of the chromatin so it can be coded. Now, there are sources of major informational molecules that are in the body associated with the nucleus. You've got the transfer RNA that brings amino acids in. You've got messenger RNA, which is read by ribosomes, and ribosomal RNA makes up the ribosomes. So the DNA is transcribed into a messenger RNA, which comes outside, and then that interacts, and so the same strand of messenger RNA is read by different ribosomes, which has ribosomal RNA associated with them, and their coding for the transfer RNA is bringing in the various amino acids depending upon the code that's in the messenger RNA. So there are different types of transfer RNAs that match different codes, and they're bringing in different amino acids to make basically the same protein. Each one of these ribosomes will be making the same protein because they're reading the same component of messenger RNA. But these are informational molecules. They transfer. They bring the code from the inside the nucleus to the outside to the cytoplasm, and they actually make the protein with the ribosomes. If we look at nuclear protein, it basically stains bluish with hematoxylin. So we can see most is euchromatic nucleus, but you can see heterochromatin in there as well. This cell here has more heterochromatin and less euchromatin. Fibroblasts, we see there another fibroblast here. These are satellite cells that support these, and these have more heterochromatin and euchromatin as opposed to a nerve cell, which has more euchromatin than it has heterochromatin. Now, if we look at the haploid genome for the different plants and animals, here we see the plants. Lily has a very big one over here. Here's mammals and humans. In terms, humans doesn't have the most of all the mammals. So there's other life forms on Earth that have many more than what the humans have, but we do have more than the yeast and E. coli. So here we see cells. These are mammal cells. These are simple columnar epithelial cells. You see the nucleus there and there with the nucleolus here and here, and heterochromatin around the edge, euchromatin in the middle. And you see they basically have a trillion base pairs associated with the genome inside each one of those cells. If we look at cell cycle, the generation times actually varies with different types of cells. In the gut, it is only 4 to 6 days before you get a complete cell cycle. In some cells, it may take for years. Most of the cells that we see are actually in interphase. They're not in mitosis. They're actually in the interphase process. During mitosis, you have also during interphase, you have a DNA synthesis for a phase there. You have a gap before that time and a gap after that time. During this time, you get centriosomes then duplicate, pull to the side, and you get the DNA starts to form threads, chromosomes that we can actually see there, and then they separate, and then the cytoplasm separates during the cycle. And we basically can see that right here. You can see the mitosis going on through here. Prophase is getting ready. Metaphase lining up. Anaphase pulling apart. Telophase is when the two cells divide in itself. In between there, you get a gap between there and symphysis. So mitosis and symphysis were two things that they could early discern. But now they're realizing more things are going on during the gap 1 and the gap 2 of the cell cycle. So this is mitosis and interphase, and most of the nuclei that we see in tissue are in the interphase. Here we see the various morphology of the cell cycle. This shows you the different times the chromosomes are put together for prophase, pull together prophase, metaphase, and then they start to pull apart, as you see there, little V's here pulling in anaphase, and then telophase, you get the closure of the process itself. This is shown by differential interference contrast or microscopy. If you remember, bright field microscopy does not you do not see cells if they're not stained. But with phased, Damarsky, and dark field, you can actually observe live cells, and this was differential interference contrast microscopy, which allows you to see live cells. Here we look at phase contrast microscopy. You can see cells are spread out on a culture plate, but then whenever they undergo mitosis, they actually round up. And in fact, they actually lose their connections there whenever they do that. So if we look at not only the nucleus but the nucleolus, we see that there is some granular portions and the fibrillar portions of there. And by cell fractionation, you can actually study the different components of the nucleolus and how it goes about to make the ribosomes. And so ribosome production is from the nucleolus of the nucleus, which then makes proteins in the cytosol when they move out there. The granular portion that we see there is 120 to 150 angstroms. It's the 45S, which is the 28S plus protein equal to 60S, a large subunit of the ribosome. So the fibrous component is about 50 nanometers smaller, and it's the 45S plus 18, not 28. 18 plus protein is a 40. It's a smaller subunit of the ribosomes. Now, ribosomes, the metabolic activity is reflected in the size of the nucleus. Here's a nerve cell, big nucleus, and a big nucleolus. The big nucleolus reflects the ability to make ribosomes. And here we see all the Nissl substance, which are ribosomes, both free ribosomes and ribosomes are associated with the endoplasmic reticulum. Here we see a Sertoli cell, a euchromatic nucleus with big nucleolus, big nucleolus here in a Leydig cell. Here it is in an egg, and here it is in a pituitary cell, a big nucleolus, indicating that it has a capability to make ribosomes on rough VR to make the granules or whatever. So here we see the nucleolus. This is the nucleolus here. Yellow here is the nucleus. So the nucleolus is inside the nucleus, as we see here. And this is the cytoplasm. So in order to study the process of the production of the ribosomes, they look for the temporal appearance of radioactivity in different portions to follow the path. They use tritiated uridine because uridine is one of the base pairs that goes into the RNA. It's what they did. So whenever they gave the cell a radioactive uridine, it at first appeared in the nucleus. And here we can see where proteins from the cytosol do go right into the nucleus. Next, it was in the nucleolus. And so next, it was in the nucleolus in through there. And then finally, it was in the cytoplasm. And so it goes from here to the nucleus, then through the nucleolus, then back through the nucleus, through nuclear pores to the cytoplasm. Evidently, this is a pretty fast trip in through there. And so you have the DNA, the genes for ribosomal RNA located in the nucleolus. You get proteins from the outside. You get recycled some of the proteins, and you make the 45S large unit, and then that here's the 5S unit coming in from proteins from the outside as well, going in there to make a big unit together, which then splits and then comes out as two different components. You get the large unit and the small unit, and finally they get together outside in the cytoplasm. So you followed the path of the production, nucleus, nucleolus, cytoplasm is where we'll see it. Here we see the big nucleolus. This is a nucleus, nucleolus, 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 nucleolus of these cells has a high affinity for ribonucleoproteins is what they have in there, so they have a high affinity for the dark staining hematoxylin. Here we see the nucleolus the nucleolus actually is different with different components of the cell cycle, prophase, different ones. It's modified. Certainly it disappeared during mitosis, but then during the different G1, S, G2 phases, a cell can have one, multiple, one, usually one big one right there is really indicative of an active cell. Here we see the nucleolus here, and in there are the genes that have the ribosomal RNA molecules are coming off and through here, and the ribosomes are being ribosomal RNA is coming up through here, and that's where the transcript is located, right in there. It's essential for protein synthesis, that is nucleolus is, because that's the source of the ribosomes, and the ribosomes, of course, read the messenger RNA and makes the proteins. Here we're seeing the plasma cell. You can see the cartwheel-shaped nucleus, spherical nucleus, and lots of rough endoplasmic reticulum in through where the ribosomes are coming here, which are the ribosomes made from the nucleolus migrating through to the cytoplasm. So remember, you can remember the contributions that are made by the cytoplasm. Proteins come in from the outside. The nucleus, you have recycling of some of the proteins. You have some of the 5S components recycling in through there. And then of the nucleolus itself, that's where you actually have the transcription of the ribosomal RNA in through there, and the combining of the different units in through there to ultimately to make the mature unit, which then slips through and gets together on the outside. So in summary, looking for a temporal appearance, if we follow uridine, it would go through the nucleus, and next it would be in nucleolus, and next it would be in the cytoplasm over the period of time after you expose the cells to uridine, and then it also used a pulse chase experiment to give it non-radioactive uridine to help follow the pathway over time. Uridine, because uridine is in RNA. Now, if we look at the chromatin, it's a double helix, as we know. It's a double helix in through there, and that's wrapped around a bunch of beads. And here we see the chromosome. This is the metaphase chromosome. There's a kinetochore, and these are the kinetochore microtubules, which are associated with pulling it apart. If we look, you can see hundreds of miles of DNA in a single cell, and if you're able to spread the DNA out, what you can see is the beads on a string. And so this is kind of the different coiling that goes on, different sizes we can see here indicating, but these are the beads on the string, and this is histamines and the DNA strands on these histamine proteins. So in chromatin, we got a heterochromatin, which is a dark chromatin, very characteristic of like a sperm. The DNA inside of a sperm is heterochromatin, dark, inactive, and then you got euchromatin, which is a light staining, uncoiled DNA associated with transcription to occur. So you have histones and other proteins from the cytosolic ribosomes that go into make the DNA and do the various components of the DNA. In terms of the messenger RNA, the messenger RNA comes down here, produces proteins in the cytosol that then go into the nucleus, as we talked about before, as well as go into the mitochondrial chloroplast and peroxisomes. So it is a cytosolic ribosomes that produce the proteins that ultimately go in to make the DNA. And here we can see the making of a protein where you have the DNA strand, you get transcription to a messenger RNA. So from the DNA, you transcribe messenger RNA, then the messenger RNA comes out of the nucleopore and interacts with the ribosomes. The ribosomes read the code. By the code, the transfer RNA matches to the different components in the code, bringing the various proteins, amino acids that match that particular code. So you get transcription, you get slicing of that, recombination of that, ribosomes bind to it, and they read the code to produce protein in the cytosol. Sometimes that protein goes back, other times it may be secretion or do other functions in there. So here we see polysomes. Polysomes are ribosomes that are attached to one another by a common messenger RNA, and some of them can be free polyribosomes, or some of them could be on the endoplasmic reticulum, or in this case, they're on the outer leaflet of the nuclear envelope. So here we have DNA in through there. You have the RNA, have the codons in through there, and then from that produces a protein. So here we have a chromosome with a certain number of nuclear genes and nucleotide pairs, and you can see you got spaces in between. You take one of those and you have introns and exons, and finally the exons go together to make the messenger RNA. So you have a longer piece up there than you ultimately have with just your exons. That's going to make the messenger RNA. It's going to be read to make the protein. Now the nuclear envelope, as we mentioned before, the outer leaflet is actually from the cell membrane, as you say, which is the same as what the raphe are on the outside. And in fact, the inner one is from the cell membrane as well. It's where it folded over itself, and that's why it's double membrane, kind of like mitochondria are. And here you see the space between the nuclear envelope is continuous with the cisternae of the rough endoplasmic reticulum that we see there. You have nuclear pores in through there, but the raphe is continuous with the nuclear envelope. Here we see the nuclear envelope with the pores. You can see the pores in through there, and the pore has a little channel there in between there, which the heterochromatin kind of pull apart as you have messenger RNA going out through there, and then also proteins that are coming in through here. Here we can see the nuclear pore again. These are nuclear pores on face view, so perpendicular to that would show you the nuclear pores that we see here. Nuclear pores, cytoplasm exchange via the nuclear pores, 40 to 100 nanometer angstroms things can go through, and there's about 2,000 nuclear pores per nucleus. Now, if we want to follow the pathway, you can actually get gold through 80 to 100 angstroms will go through these nuclear pores. Nuclear plasmids are chaperone proteins that chaperone proteins from the cytoplasm into the nucleus, and so they bind to things. It could be the proteins, it could be the the deoxyribonucleic acid that's coming through there, and you can see that each of them have a head and a tail part, and it's actually the tail part that's involved. If you have radioactive nucleoplasmid, then they will go into the cytoplasm and then to the nucleus. If you just cut the heads off with limited proteolysis, then what will happen is that they will not go into the nucleus. If you just use the tails themselves, they will go into the nucleus. And here you can see where they took immunogold, they put gold, not immunogold, just gold particles, and they attach the radioactive tail part, and that was able to take the colloidal gold, into the nucleus so they can see what size gold could slip in through the process. Now, the nuclear pore is a certain size, however, 40 to 100 angstroms. However, the large subunit is 120, 150 angstroms. How do you get it through? Well, what happens is the subunit kind of unfolds and makes itself linear so it can go through the nuclear pore and a smaller nuclear pore. Here you can see the nuclear envelope, and this is where one of those subunits is squeezing through the nuclear envelope, kind of as we see here. Here's the nucleus getting out to the cytosol, as you can see there. So we have a nuclear pore. The opening is kind of about 9 nanometers in thickness, and here you can see again where you got your gold coming through there, and the pore size, some things can come through. Here you see an on-shot view of them. You see there's different granules in through there, but these can open up to let things go through. Here we see the nuclear pore, and just inside the nuclear pore is the nuclear lamina or the fibrous lamina, and that's intermediate filaments, like you have intermediate filaments on the outside as well. Now, nuclear pores, DNA is not associated with nuclear pores. You can't lose your DNA in the process, but you do want messenger RNA to come through. So RNA is associated with nuclear pores, but DNA is not. So here we see the fibrous lamina or nuclear lamina is through there, intermediate filaments that is there. And some things can just go through the nuclear pore based on the small size. Bigger things have to be actively transported across it. Sometimes, as we mentioned to you, the nuclear pores that we see in the nuclear envelope, the nuclear envelope is continuous with the rough endoplasmic reticulum, and sometimes you can see things that look like nuclear pores in the plasmic reticulum there. We call it annular lamellae, or nuclear pore-like structures in the rough endoplasmic reticulum, where these could be a reserve, not sure exactly what it is, but they look like nuclear pores, and indeed this cisternity is continuous with that space in between the two components, the membranes of the nuclear envelope. Now, Y chromosomes, kinetochores, and centrioles. Centrosomes, which are really two centrioles together, are supercores. They prevent errors in mitosis, is one of the reasons. Chromosomes are supercoiled DNA, and they prevent problem with it. That's why we have the chromosomes here. They're all tight and tucked so that they don't have mistakes. Now, kinetochores are proteins that the microtubules are attached to them, and that's the most important part. If you cut the chromosome, it won't migrate, but as long as you have the kinetochore associated with it, it will migrate. Many plants don't have a centriole. How they do it, I don't know. If the centriole is removed, the cells can still divide. The real function of the centriole is not known, other than it's known to have labile microtubules radiating from those, and it's important to be passed on to the daughter cell for whatever that function is. Here we see microscopy allows to observe live cells, differential interference contrast microscopy. Here is dark is phase contrast, and we see the centrioles are located here, and the chromosomes are there, and microtubules connecting to there are there as well. Now, why do cells divide? They want to increase their mass, to reduce the damaged cell, and to reduce the size of cells to increase diffusion. If you have a small cell, you could increase the diffusion by not having the source of nutrients or oxygen far away from the center of the cell. Also, to avoid aging, to build up waste, and here you can see where cells are dividing and waste is isolated to a certain cell, and you can get rid of that cell to prevent that. Now, mitosis is defined as cell division in which duplicated chromosomes divide into daughter cells that have identical chromatin content. So you don't want any change. You want to have the daughter cells that have the very same chromosomes and genome as the parents. And that's what we have here. We see some mitotic figures in the gut. And in the gut, it's really in these crypts. So these are intestinal villi projecting in little crypts in through there, and that's where you have the mitotic activity going on, rapid division that goes on. And then as the cells differentiate, they migrate up to the surface and then are sloughed off. The karyokinesis is nucleus division, and so here we see a metaphase plate. There's a mitochondria in through there, and we can see where some have been removed, pulled apart, new cells developing here, cells in mitosis in the intestines, what we see. So karyokinesis is actually a separation of nucleus. Cytokinesis is where you have separation of the cells that you see here to produce two daughter cells. Now, whenever a cell gets activated for cell cycle, you get some kind of growth factor or some kind of factor interacts with a receptor, a second messenger inside there, activates kinases, different kinases in through there in the cytoplasm, and then it goes into the nucleus. It activates the genes that regulatory proteins, early response gene, delayed response gene. Ultimately, you get activation of the cell for the cell to divide. So chromosomes are like rod-like structure organelles during the cell division is what we have. Diploid is 2N, full complement. So there's two components, one from the parent, from each of the two parents. Haploid is half the number. That's what you have for gametes in the egg or in the sperm. And polyploidy is where you have more than the complement. For example, Down syndrome is when you have too many chromosomes. You get duplicated chromosomes there. And so the chromosomes or rod-like organelles are visible at the light microscopic level. You can visualize those. Now, a gene is a hypothetical unit of inheritance. A linear range along the length. It's got gene locus along the depletion maybe could occur. Translocation occurs along the length. So this would be a nuclear gene right in through there, of which ribosomal RNA is being read off of this. Just happen to be in the nucleolus. The three main units, events in mitosis. You have orientation. At the chromosomes condense during prophase. The nuclear envelope breaks down, and then they align in metaphase plate, and then the anaphase, they start pulling apart, and you have segregation. So you have orientation, alignment, and then segregation, and then the final part is separation of the cytoplasm itself. In mitosis, here we can see various things in mitosis. These are the prophase in through there, metaphase, in through anaphase, pulling apart, early anaphase, later anaphase, and then you can see where these are pretty much pulled apart right in through there for telophase. So we see the different phases that's going on. Again, prophase, you got breaking down a nuclear envelope, and then alignment, metaphase, anaphase, pulling apart, telophase, is separation of the cytoplasm. So, as I mentioned to you, you got duplication of the centrosomes that we have here, and they move to the different poles. And then during prophase, you have chromosomes are lining up, and they're lined up in metaphase at that point in time because there are different chromosomes, different microtubules are acting on them, and as they pull and push, the forces equalize and on the metaphase plate, and then they pull apart. So you get orientation, you get alignment, and then you get segregation to occur. As I mentioned, the nuclear envelope is broken down. So the nuclear envelope is broken down during the process, and then after telophase, the nuclear envelope is reestablished. And so we have this. So in one sense, it kind of goes back to a more primitive state in that it has no nucleus at this point in time, but then the nuclear envelope fits back. And so during a telophase, you have the two groups pulled apart, and then during cytokinesis, you have the actin ring around the center of the double cell, and that squeezes down until you have two different cells develop. And here we can see them. They're pulled apart, get microtubules running back and forth, and then you have actin filaments that squeeze that down, that pulls these apart. You can see the actin squeezing down, actin-rich area right into this area. You can see with the fluorescent staining, the rich actin area in the middle, this being a nucleus, and a nucleus here of a cell that's undergoing cytokinesis. Again, we can see the squeezing of the cell, the hourglass that you can see it, and then finally the two cells have been complete. This is a scanning electron microscopic view of the process where two components, a double cell, is dividing in the middle, and you can see the cleavage furrow right there that's going to separate to two different cells. So the centrosome separates, duplicates, and separates, and then pulls the chromosomes at the same. This is G1. It's actually during the symphysis phase that you get duplication of the centrosomes. During prophase, they're pulling apart. They have these aster microtubules that come from them, and then they are associated with chromosomes pulling apart, and then finally one ends up into the different cells. Here we can see where I got from the internet. This would be the centrosomes here and here. The pink is kinetochore microtubules, and then these are kinetochore attached kinetochores, which is where the chromosomes are attached to the kinetochore microtubules, and then you have the aster microtubules running out through there. You can see it right there. The green, are the microtubules. The blue is the chromosomes, and the kinetochores are the pink. And so there's basically two models for pulling the chromosomes apart. One is that you have the microtubules attached to the kinetochore, and you have ATP-driven chromosome movement drives the disassemble. So as the thing moves along, it disassembles, so the path moves along after it. And then you have microtubule disassemble driving the chromosome movement. So in one case, it moves along the kinetochore and it disassembles afterwards. So either disassemble makes it move, or it disassembles after it has moved through. But in either case, the microtubule is disassembled in the process of movement. And this is moving in a direction of it. And here we can see microtubules. These are tubulin going to make microtubules, and how they can disassociate. As they disassociate, the protein associated would be moving down, pulling the chromosomes with them. And so here we can see the centrioles sending out different types of microtubules. There's basically three types of microtubules. You've got the aster microtubules that project out from the centrosome. Also, you have the polar microtubules that are interacting with the poles, and then you have the centrosome spindle pole as well. And so basically you have kinetochore microtubules, you have polar, and you have astromicrotubules. Those are the three groups. So here's the kinetochore ones that actually are connecting to the kinetochore. These are probably the ones we're calling polar. Polar microtubules here go to, I don't know what the difference is. This is actually connecting to the chromosome. The polar one actually interacts with one another, and then you have the astromicrotubules. So you have astromicrotubules, which actually are on this side. The polar microtubules are going to push against the other polar microtubules. The kinetochore microtubules, it's going to shorten, and that's going to pull that part of the chromosome to the different one. And the asters are going to pull it further apart. And so here we see the chromosome. This is a kinetochore in through there, and these are the kinetochore microtubules that are going to pull the chromosome in that direction. So the chromosome movement, you have an imbalance of powers between the different ones, and whenever you equalize the power pulling and pushing, then you're going to basically be on the metaphase plate. So you have shortening of the kinetochore microtubules, which pulls it along. You can see where this is shorter here than it was over there. And then you have the polar microtubules are pushing one another apart. So they're pushing the poles apart. The astromicrotubules are pulling the centrosomes apart as you're through there. And then you have the third part is the shortening of the kinetochore. So the first, push the poles apart, pull the centrosomes apart, and then shortening of the kinetochore microtubules that brings it along. So those are the three parts of it. So you got the polar ones pushing, ester ones pulling the poles apart, as well as kinetochore shortening, pulling the different components of the chromosomes to its respective size. So here we see kinetochore microtubules, polar microtubules, and astromicrotubules located there pulling the chromosomes apart. We can see a lot of mitotic activity in an egg. You can see these are dividing cells, metaphase plates that we can see. Next time, we're going to talk about peripheral blood is what we'll do. Either that or we're going to go to South Dakota. This may be in North Dakota, but right over there is the Black Hills of South Dakota. And what's on those hills? There's Mount Rushmore, where you have the presidents located. Also located pretty close there is the mammoth site, where there's a lot of mammoths that were associated with there. They were digging it up, and they found that there was big bones there, so the guy with the bulldozer stopped, and then they took that area and made it into a museum. So we want to thank various books, including Bruce Albert's books, Molecular Biology of the Cell. A lot of pictures were taken from those, as well as other textbooks and a couple from the internet as well. We want to thank those for their contribution to education. As I did not make these, I want to acknowledge they are ones that had these ideas of drawings and images which tell the story of the molecular biology of the cell. Thank you.